My name is Nicola Longo. I'm a medical and biochemical geneticist at the University of Utah in Salt Lake City. I diagnose and treat children with inborn error of metabolism and genetic condition. The most common type of children that I see in my clinic are children with intellectual disability, and we try to develop strategy to identify what caused their condition, and also to see if we can help in treating the cause of their problem by identifying the specific gene that it is causing the genetic syndrome.
What types of genetic testing are available to families who have children who may have concerns?
The type of genetic testing available to identify the cause of genetic syndrome continues to evolve. In general, the first test done to children who are behind in development, if we cannot identify a very specific genetic syndrome based on clinical exam, is a cytogenomic microarray, which is a test that determines whether there are large deletion or addition to the genetic complement. In the old day, we used to do a chromosome study. The cytogenetic microarray is a cytogenetic study. It is like a chromosome at a much higher resolution, capable of identifying even very small deletion or duplication. The additional thing that it is done by the microarray today is the identification of whether both copy of a child chromosome are derived from the same parent or different parents. And this is done by looking at what we call single nucleotide polymorphism, or SNP. And in general, these are very different among different people, for which normally the two chromosomal copy have different type of single nucleotide polymorphism. If they are identical, then it could be possible that both copies of a chromosome are inherited from the same parent, or that the parents are related to one another for which they share genetic material. This technology identified the cause of delays in development in about anywhere between 10 and 20% of affected children. In general, the likelihood of identifying a genetic cause is higher if the child has severe symptoms, and many times if there are associated alteration, physical alteration in some cases. If we cannot identify the cause of the delays in development of the child, then we proceed with additional testing. In the past, everything was based on clinical examination, the suspicion of a genetic syndrome, and the sequencing of a specific gene. Today, we have the luxury of being able to sequence all genes in the human body. And we do that by whole exome sequencing, which is a technology, it is a blood test, in which we take the DNA from the child, we compare the DNA of the child to the one of the parent to identify if there are de novo changes. De novo changes mean that neither parent has a genetic variant identified in the child, and many times the new genetic variant can cause significant medical problem in the child. And then if there are not the new genetic changes, we analyze all genes in the human genome to determine if there are variants in genes that we know are associated with the medical problem that we see in the child. And then we make a call to determine whether those variants are the ones that cause the symptom in the child. The testing that we do today is called exome sequencing, in which we only sequence a portion of the genome. So in other words, there are portions of the genome of which we do not know exactly what they do, and we do not sequence those. The disadvantage that obviously there are variants in those regions as well that could cause the child problem. We are working in that direction, and many centers now start doing whole genome sequencing, so they sequence all regions of the DNA. And the advantage of whole genome sequencing is dual. Number one, you can identify deletion and duplication like the one identified in cytogenomic microarray, deletion and duplication that many times are not visible in the cytogenomic microarray. The second thing is that you sequence regions of the DNA that are normally not sequenced by exome sequencing. As a result, you have a full coverage. The disadvantage is that the amount of information can be overwhelming to understand and to interpret. And sometimes we are still left with the question of whether the cause of the child problem are genetic or might not be genetic.